We've heard it all before. The mitochondria is the powerhouse of the cell. The nucleus is the control center, and the cell membrane determines what comes in and out. But what does that have to do with you, a future nurse or doctor, and how does that relate to our anatomy and physiology class? I mean, aren't there enough complicated structures that we need to learn about already? Actually, the cell biology is the most important lecture in all of the series of anatomy and physiology because you are made of 30 trillion cells. And if you are doing something, it is because your cells are doing it for you. So in order to understand how your whole body works, we need to understand how these cells work. Hey everybody, Organized Biology here, where we make difficult biology concepts simple. And today I want you to forget everything you learned about back in high school biology class, because they showed you this picture of a cell and they pointed at all the different organelles and they told you what they did, and they were taught in a vacuum and you had no idea why you had to learn about it. Well, today we're going to go through several different topics of cell biology, you can see on the top left of the board, but we're going to apply them to different scenarios in your body. So let's get started so you can be successful in your class and in future healthcare. So first off, when we look at a cell, I don't want you to think of the cell as just a block that has stuff in it. I want you to think of it as very specialized. You know, you're made of 30 trillion cells, and there are what? 8-9 billion people in the world. We don't all look the same. We don't function the same, right? We have the same bare bones and stuff like that, but we're made of different things. Same too are our cells. I mean, look at the different shapes and sizes and colors of these cells. As an example, this guy right here is called a cardiac myocyte. Cardiac myocytes are basically the cells, if I can spell right, are the cells in your heart that help to contract. So they've got these lines and these proteins that help accordion those guys closed so that they can contract properly and eject your blood. We've got these neurons here, right? These neurons are in your brain and your spinal cord and your nervous system, and these neurons can be up to 3 feet long and sending signals. Very different structures than this. This is a fibroblast, a pseudostratified columnar epithelial cell. This is an erythrocyte. I want to point this guy out. This is your red blood cell. So all of these different cells have different structures, and we know if they have different structures, they have different functions, right? And now what I want to point out quick is you notice site is a very common suffix. Anytime you see site in A&P class, it just means cell, okay? So for this guy, this is the heart myomuscle cell, and this is a red erythrocyte cell. It's a red blood cell, okay? So anytime you see that suffix, know that is what level we're talking about, okay? So I would venture to say the most important structure of the cell is going to be that membrane, the outer part of the cell. Now, key with the membrane is that we are floating around in this fluid-like environment, right? So your body is mostly made of water. We know that, right? So there's going to be a lot of water out here in this space, as well as in this space. Well, what are these three spaces that we're talking about? You probably know this one, right? This is your bloodstream. In your blood, I want you to remember this core concept that the blood is a river of life, and it goes to virtually every cell of your body. So 99% of your cells are going to be bathed with this blood. Now, the blood has some fluid-like components in it, like with water and some other electrolytes. Maybe you've heard of them, sodium and potassium, as well as nutrients like amino acids and glucose. We'll talk about them. And some of those components can actually leak out into that space, right? So your cells are being bathed by some of this stuff from the blood, right? Now, this is called specifically the extracellular space. Extracellular space, you may also hear it be called the interstitial space. Interstitial, I believe, means in between, inter, two places, stitial, okay? So you can see we've got fluid in the blood, we've got fluid in between the blood and the cells, and then obviously inside the cells, we call this the intracellular fluid. Intracellular fluid, very cool. Now, how do we separate these spaces out? Well, obviously you see we've got the cell membrane itself, right? Separating the cell's inside environment from its outside, as well as in the bloodstream. These are actually very thin cells, the red lines that I drew. So we've got these separations of spaces, okay? Now, we know that the membrane separates it out because the membrane is mostly made of a lipid. So I'm going to draw in red right here. These are the lipid aspects of the membrane. Okay, so we see that the membrane is mostly in space made of lipids. So we're just going to say the membrane is a lipid just to make things easier. That'll make sense in a little bit. Now on the outsides, right, on both sides of the lipids though, we have these very, very small charged phosphate molecules. small and charged phosphate molecules. Now, why is that important? Well, phosphates are charged, as I said, and anything that is charged, like a polar molecule, you may have heard of that in a previous video, that can associate well with our friend water. Water likes polar molecules. But then, since we've got a lipid buried inside of it, it separates it out so that the water can't really pass through the membrane, nor can any other polar thing. So we have the separation of environments, okay? Now, that being said, we need sometimes to bring things in to the cell and sometimes take things out of the cell. It's called cell transport. Let's talk about it for a little bit. We're going to need some things to get into the cell. I'm just going to straight up tell you that. Two such things are going to be coming from the blood, like lipids. Maybe you may have heard of this one before, like cholesterol. These things will be carried around in the blood with some carrier proteins, as well as you may have heard of this molecule before, oxygen. Very important gas molecule that you breathe in. Both of the times when you eat fats, when you breathe in oxygen, it goes into your bloodstream. Now, we need these things in the cells for a variety of reasons, and I can just simply draw them going like this. This is so easy. Any lipids and any gases like oxygen can just pass right through the membrane. Why is that? Well, it's because the membrane is a lipid, and we know that lipids are called non-polar molecules. They don't have a separation of charge inside of them, so therefore other non-polar molecules like gases and lipids can just pass right into the cell through the membrane very easily. We call this process simple diffusion. Okay, you may have heard of that before. Now, simple diffusion, simple because there's no extra need for anything like a protein, and diffusion because things are moving from relatively high concentration to relatively low concentration inside the cell. So I am assuming that in the cells we have low oxygen and we have relatively low, in this case, cholesterol. So we're needing some cholesterol in the cell. So therefore, these things can just pass from high to low concentrations. Very easy, okay? So that's one core concept, is that things in nature like to flow from high to low. There's no energy required. Now, that being said, we are smart. We've learned some of these things before, and we also know that the cell needs several different other things. So I'm just going to write needs inside of the cell. One such thing is glucose. You've heard of glucose before. This helps us for ATP production. Yes, we're going to learn about that in a second. We also need things like amino acids. Amino acids are your monomers of proteins. Monomers of proteins. So in other words, they're the Lego blocks that help build the proteins, and the cell's made of proteins. We'll see that once again in a second. We also need several other things, but we're just going to focus on these two for now, okay? Now, the problem is that glucose and amino acids and other things are polar. That means there's a charged aspect to these molecules, so polar things cannot just pass in and out of the membrane easily. In order to get in and out of the membrane, we actually require things called proteins, okay? See, proteins, they're important. So in the membrane, I'm going to draw this real quick. We have a very, oh golly, so very important protein that we call the sodium potassium ATPase pump. We have so much to break down for this. It's so important. You've probably been taught this before and you were like, this is the weirdest protein I've ever heard of. Why is sodium getting thrown out or potassium getting thrown in? This runs your body. In fact, if you eat any food, 60% of those calories is estimated to go towards firing this one type of protein pump. So it must be important. So what is this protein doing, and how does it help us get these things in? Well, stay with me. Check this out. First off, ATP. ATP is going to be our cellular currency for the cell. We're going to talk about how we produce it later on, but just know that it is necessary to do a lot of different cell functions. So ATP, very important, cell currency of energy. That ATP is going to fire this thing called a sodium potassium pump. We're going to draw ATP. Going into it, we're going to clip off the last phosphate of ATP, and we're going to release some sort of usable energy, okay? If you want to learn more about that, you can watch my ATP video. Now, when we do that, what happens is that we are going to pump sodium ions out of the cell. So we're pumping sodium ions out of the cell. So I'm going to draw sodium coming out, and then I'll draw potassium in a different color here. We're going to draw potassium coming in, all right? So here's potassium coming in. If you want to know, it's 3 sodium per 2 potassium. It doesn't matter. Now, what that does is it tells us that if we're firing this, we're going to have relatively high concentrations of potassium inside of the cell and relatively high concentrations of sodium outside of the cell. You see that? So we have this concentration gradient. And what's really interesting is you can memorize this and know that in the blood itself too, there's going to be pretty high sodium and relatively low potassium. And on the flip side, as we just mentioned, there is going to be relatively, did I just draw that in a different color? I am colorblind as can be. Or is that brown? I can't really tell. Well, we're going to figure it out. Comment below if these are the same color. It's terrible. Therefore, we got low potassium out here, high sodium out there, and then we'll also have low sodium in here. That's what we can assume from this. Okay, that's because the sodium-potassium pump's always firing. So if I were to ask you, where does sodium want to go? Well, obviously it wants to go into the cell. Or potassium wants to go out of the cell because it wants to diffuse from high to low, as we mentioned before, right? Things in nature like to flow from high to low. But why can't they go in and out? Well, because these are polar as well. So these things are called electrolytes, charged ions like sodium, potassium. We'll also have calcium in relatively high concentrations outside of the cell. So I'll write calcium out here and actually relatively low calcium in here. So all of these are electrolytes in differing amounts, right? But they can't get in and out because of that nonpolar membrane. So all of these things need some help getting in and out. So once again, how do we get these other things into the cell so that they can build proteins and make ATP? Well, let's check this out. Since we have fired that pump, we are now having a concentration gradient that can fire other proteins. So let me draw this other protein, maybe in another color. Well, we'll just do blue. Okay, so here is another protein that I'm going to draw in, and it's going to look like this. Okay, so there's going to kind of be one aspect of it here, and then maybe I'll draw a different aspect of it right here. What we call this is a symport protein. So I'm going to write symport here. And this is a very common protein. Symport means same direction of transport or same transport, okay? Now, that means that this protein is going to move two things in the same direction. Those two things are going to be, I'm going to write sodium, and it's going to want to throw sodium this way. And it's also going to be either, it depends on the protein, it could be a different type of symport. It could be a glucose, or it could be, you could probably guess, amino acid. So there are some of them called sodium glucose symports. There are some of them called sodium amino acid symports, all right? Now, what can you predict? Where will they pull glucose and amino acids? Also into the cell. Now, why do these work? How do these work? Well, we know that they're going to bring two things in the same direction in. But why does sodium want to go in? Huh? Because of that sodium-potassium pump making it really high outside. So what's going to happen is sodium's going to see that symport and be like, ooh, I want to run in. I want to run in. And it will. But it can only run in if it has a partner. Who's the partner? One of these two. So we'll just say in this case it's going to be glucose. So I'll draw glucose as this little hexagon. That glucose is going to fly in as well. So check this out. We are bringing in our glucose. We are also bringing in our sodium. Wonderful. Now, why is that important? Because later on we'll talk about how glucose helps us to produce about like 32 ATP molecules. That's a lot. So just by having a gradient of sodium high outside, pulling glucose in, we can now use that glucose to make ATP and keep this process going and going and going. How amazing, right? So those are just some types of proteins in the membrane. There will also be several other types. I'll just point them out real quick. Some of them will be called channel proteins. They're always open, allowing things to flow in and out. So as an example, if this is, say, a sodium channel protein or a leak protein, this sodium would constantly be able to leak into the cell. Whereas if it was like a potassium leak protein, potassium would be flowing out of the cell, right? So it's always open. There will also be other proteins that will look a little different. They might be like this and maybe have like a long chain, perhaps, of glucose. So like a big long chain of a carbohydrate on the outside. These guys are going to be called glycoproteins, okay? And these glyco, sugar, proteins, so there's a sugar part and there's a protein part. A lot of the times, these serve as ID tags for the cells, ID tags. This is how we know if our cells are us, like human cells versus bacterial cells. There will also be some other proteins that will be voltage-gated proteins. Okay, so they'll have a charge in a way associated with their structure, and we'll also have things called ligand-gated proteins. So let me draw one here, a ligand-gated protein. Okay, now let's look at these two. This one is a type of channel protein. It can open up, but only when a specific chemical is secreted, right? So we can bind that, and then this guy will open right up. So that is what these proteins will be, ligand-gated channels. I'll write that, ligand-gated channels. So that means ligand key into the lock to open it up, and it could be like, say, a ligand-gated sodium channel. It'll open up when that chemical is here and allow sodium to rush in. This is actually what happens when you contract your muscles. So if you had, for example, a skeletal muscle cell, okay, and you wanted to tell that skeletal muscle cell to contract, you would actually have one of these neurons, I'll draw in orange, that would release a ligand. We'll draw it as that little purple. That neuron releases the ligand, okay? It will bind, as I've mentioned, to that receptor, that sodium-gated channel, ligand-gated channel, okay? It'll open up, and what will happen? Well, in this case, sodium will flow into the muscle cell. That makes the muscle cell very positively charged, and it actually tells the cell to contract, okay? Now, at the same time, it got charged, right? What are we talking about there? Well, cells in general are going to be made from, as you're seeing, a lot of proteins. So I'm just going to draw a few other proteins in this cell, and we're going to talk about where they come from later on. So just know inside of the cell, we're packed full of these very important proteins. Some proteins serve as like the cytoskeleton components, like they're holding the cell up so that they keep their structure and their function. But notice how I'm drawing them. These proteins have a strong negative charge associated with them, right? So that means that inside the cell, if I were to like put a voltmeter and measure the voltage of the cell, the voltage would be somewhere around -70 to -90 millivolts, okay? It's a good number to remember. We call this cell's voltage the resting potential of the cells. It's at rest when it is negative. But as you can see, when it's negative, it's at rest, not doing much. But when it gets positive, it can do cool things. How do we get positive? Well, we let in positive ions, or we could actually lose negative ions as well, but we won't get into that. Now, why does that become important? Well, let's say this cell gets to, I don't know, -55 or something. So it gets to a certain voltage. Well, that's where these voltage-gated channels can open up. So voltage-gated channels. These guys open at a certain voltage. So say the cell bumps up to, say, -55. These guys pop open and allow different things in as well. So you can see there's a variety of different membrane proteins that will close and open at different points in time to help the cells function. As an example, like the muscle cell. Beforehand, the muscle was negatively charged, not doing anything, but when the neuron told it to contract, it sent that little chemical, opened up the ligand channel, sodium rushed in, made the cell positive, and the cell contracted. So it's important to be able to regulate the things coming in and out of the cells, and most of the time, it's conducted by proteins. Proteins, I'm saying this word so many times. What the heck are they and how do we make them? Well, we know inside the cell, in most cells, we have a big dark spot. And we know that this big dark spot is called the nucleus. And inside the nucleus, we know, is a big, long strand of cool molecule that we call DNA. Now, let me be very careful with this. We know that DNA will eventually code for proteins, okay? That's an easy thing to memorize. It's a harder thing to understand why and the steps. So let me say first off that the DNA is basically the same in most cells. What does that mean? That means that the same instruction manual in, like, say, your skin cell is the same instruction manual in your heart cell. But they look very different, right? So like your heart cell, your fibroblast cell here, your neuron cell, they all have virtually the same exact instructions, but you know that they don't look the same, right? They all have different functions. So how did they get different structures? Well, know this. Cells only read the parts of the DNA that pertain to them. That's a very important point. Pertain to them. What does that mean? Well, let me use an analogy. Let's say you have a big recipe book that you're opening up at 6:30 p.m., okay? When you open that recipe book, are you going to open to like the breakfast section? Well, no, you're going to open to the dinner section and make dinner for your family, right? So you only open it up and read the sections that's pertinent to what you need to do at that moment in time. Well, the cells are smart. They only read the certain segments of DNA that code for their specific proteins, their specific structures. These specific segments are going to be called genes, okay? So think of genes as the little chapters of DNA, okay, chapters of DNA that code for proteins. Okay, so that is very important. So if I'm taking this example, this cell at one point read the DNA to produce all of these different proteins that are embedded in their membranes, right? So how did that work? Well, we know that DNA has a code inside of it. That code we know is those A's, T's, G's, and C's. A bonds with T always, and G bonds with C. As we read them, we transcribe them onto a molecule that we call mRNA. So I'm going to draw a little mRNA molecule here. mRNA molecule right here. That's going to be our transcript. So we read the DNA, we read the letters, we make an mRNA transcript. That mRNA can actually leave the nucleus because it's thin enough, small enough to fit through these little proteins in the nucleus called nuclear pores. So we've got the transcript now out of the nucleus, and we're now into what we call the cell fluid, the cytoplasm. So we've got that mRNA transcript. As I mentioned, that mRNA is going to have the code essentially for how to build a protein. Yes? Now, how do we build these proteins? How do we know what structures to put them in? They're coded on this mRNA, as I mentioned. So this mRNA will go to a structure in the cell. I'm going to draw a few of them. And these are going to be called ribosomes. You may have heard of them before. Ribosomes are the centers for producing proteins. So think of them as little chefs that will read the recipe to produce the proteins. Now, as that happens, so mRNA goes in, goes to the ribosome, and then the ribosome is going to produce the protein. Now, the thing is, where do we get the base units for these proteins? How do we get the base units to form all of these types of proteins that I mentioned? We've already talked about it, y'all. Look over here. We got amino acids in from the bloodstream. Yes, because how did we do that? Well, we know that we eat food, we break it down into carbohydrates like glucose. We break it down into amino acids that I haven't drawn before. We'll draw it in purple as well. Here are the 20 different amino acids that we've eaten from our food. And how did they get in? Well, they used those sodium amino acid symports fired by the sodium potassium ATPase pump that then pulled it in. So now all of these amino acids that are in the cell can come over here and be used by the ribosomes to build that protein. So in reality, the protein would basically look like a bunch of amino acids that are chained together in such a way that form these structures that we've been pointing out. Now, it's kind of hard to draw that, right? But just know that amino acids build the proteins. They're like little Lego blocks, and we need to place them in the right order to form all of these different things, proteins, that help the cell's structure and function. So you see how, man, cell transport comes into play. Protein synthesis comes into play, all to make these structures, all to help the cell's functions, as we mentioned with the muscle cell previously, right? So these are so important to understand. One connection to this is genetic disorders. Let's say you have a genetic disorder, genetic disorder, where you have a gene that is incorrect, okay? It doesn't have the right code for the right protein. As an example, let's just say this hypothetically. What if you had a deficient gene, so a gene that had the wrong code, you couldn't make that protein for this specific ligand-gated channel that was in your skeletal muscles? If you couldn't read that right DNA, you couldn't make the protein. If you can't make the protein, you don't have any receptor, right? You don't have any receptor for the neuron signal. And if you don't have a receptor, you don't change your function. So therefore, this skeletal muscle would never be able to get positively charged. Your skeletal muscles would remain negatively charged. And if they did that, would you be able to contract your skeletal muscles? No, you wouldn't be able to move. So do you see how, man, in a very real way, a genetic disorder is when you have a gene that is incorrect. Maybe you inherited it. Maybe you had a mutation. We won't talk about that. And if you get that, well, now you can't code for a protein. Well, the protein's important for a function. If the functional thing is not there, you can't do the function, okay? So really, really important to understand how that works. All right, let's move on. We need to talk a little bit about ATP production. ATP production. First off, I'm going to erase a little bit of this just so we have some more space. I appreciate you guys sticking with me. If you've enjoyed this so far, please like the video, subscribe to the channel. I really appreciate you guys' support. It means the world to me, seriously. Like, this is such a blessing to be able to teach and hopefully make it a little helpful, right? So you can succeed. Now, ATP. Probably heard of it before, and it is standing for adenosine triphosphate. So what that means is there is a molecule called adenosine, okay? It's a nitrogenous-based molecule. And then we've got 3 phosphates attached to it, 3 phosphates attached. Now, this molecule is vitally important because that last phosphate contains a lot of potential energy. And when that phosphate gets cut off, that phosphate goes flying, okay? So it goes flying around, and it does some sort of useful work for the cell. So then we are left with ADP, adenosine diphosphate versus triphosphate, and an inorganic or by itself phosphate. Where have we seen this be used? Well, the sodium potassium ATPase pump. We had an enzyme in ATPase that clipped that phosphate. Phosphate went flying. What helpful work did it do for the cell? It threw sodium out, it threw potassium in, so we can fire all these processes. So we call ATP the cell currency of energy. Now, the question is, how do we produce it? And how do we go from adenosine diphosphate, right, after we use ATP, and how do we produce ATP again? Because it's vitally important. How do we reproduce ATP? Wonderful question. Well, we need several different steps. So the first thing we need is a molecule of glucose. Glucose is going to be our fuel source. So here's glucose. Glucose is going to be our fuel source. Why is it a fuel source? Well, it's a big molecule. It's got 6 carbons, 12 hydrogens, 6 oxygens, a lot of bonds there, so there's a lot of energy. And we also will need oxygen. So glucose and oxygen, specifically 6 of them. We're not going to go through the details. Now, what happens first, I mentioned that glucose is a 6 carbon molecule. So I'm going to draw 6 circles, and these are all going to be carbons. So I'm going to draw a little C inside of them. Okay, so we're going to hypothetically say that this is that molecule of glucose. Well, we know that anytime we break bonds, we will release energy. Okay, so what happens first in the cell to make ATP, so don't lose the forest for the trees, we're trying to make ATP, we're going to cut that molecule in half. This process is literally called the sugar breaking process. Sugar, lysis, to break. So we're breaking that sugar in half. So now we have 3 carbons in each molecule. So here's a 3 and here's a 3. These 2 molecules are called pyruvate. Okay, so pyruvate molecules. These guys, fascinatingly, will find their way into your favorite organelle. Here's your favorite organelle, the mitochondria of the cell. So here's the mitochondria. I'm going to write mitochondria. And this mitochondria is the powerhouse of the cell. It's the site of ATP synthesis. But what that pyruvate will do is find its way into the matrix of the mitochondria. So we've got these pyruvate molecules in the matrix of the mitochondria. Now, what's going to happen in here? Well, in this section, we are going to do something called the Krebs cycle. You also might hear it as the citric acid cycle, where we're going to further chop the pyruvate up, okay? And we're going to steal some of its hydrogens, and we're actually going to release CO2 in the process. That's how we actually produce CO2. But when we chop them all up, we form these molecules called NADH and FADH2. What do we see here? Well, they are carriers of those hydrogen atoms, right? So we've got hydrogens in these molecules that we stole really from the glucose, from the pyruvate in this process. We call these guys, you'll see this in your textbook, as electron carriers. Electron carriers. Well, what happens next? Well, the electrons are being carried. They're hidden in the hydrogen atom. So if you need this review, you've got a hydrogen atom, you've got a proton, you've got an electron on the outside. So if we're carrying a hydrogen atom in here, right, in these molecules, therefore there's an electron in them, and we're going to carry them. Where are we going to carry them? Well, we're going to carry them to the inner membrane of the mitochondria, where we have a process, a protein process called the electron transport chain. Electron transport chain, where we're going to quite literally transport electrons through this chain of 4 proteins. There are actually 4 proteins here, and there's one on the top too, so I guess there's a 5th. Let me ask you this real quick. Where'd the proteins come from? Well, the process of protein synthesis, right? So this is also vitally important in terms of energy production, protein production. And so we carry these electrons here, okay? And those NADH and FADH2 will come to that area. What they will do is donate an electron, right, to the electron transport chain. That takes away the electron. That keeps the proton, the hydrogen ion, H+, and they keep the H pluses in this intermembrane space. So we're going to build up this intermembrane space with a variety of hydrogen ions. When we do that, what's fascinating is the electron's going to fly through that electron transport chain and actually get picked up by our friend oxygen. Oxygen is going to serve as the final electron carrier. So here's our oxygen, and that is going to catch hydrogen in a second. We'll see that. And that hydrogen, what's it going to do? Well, it's going to flow through this amazing molecule called ATP synthase. So let me write that here, ATP synthase. What does that mean? Well, it's an ATP synthase. That means a builder enzyme. It's a builder enzyme that's going to produce ATP. The way ATP synthase works is it uses the gradient of hydrogen, the high hydrogen in here, and it allows that hydrogen to flow through itself. And in the process, it will staple, catch this, ADP back onto that phosphate. So we go from, I'm going to erase our electron carriers here. We go from when hydrogen flows through, ADP, the phosphate will actually be attached here. And when that hydrogen flows through, we're stapling it back on. And that forms our ATP molecule. How cool. So once again, just to rerun you, the electron carriers carry it to the electron transport chain. The electron gets thrown through the electron transport chain, picked up by oxygen. The hydrogen gets thrown out into this intermembrane space. That hydrogen flows through ATP synthase, high to low, powers the ATP synthase to staple phosphate back onto ADP, forming ATP. Now the question is, where does that hydrogen go? Well, the hydrogens will bind to our oxygens. When we bind our hydrogens to our oxygens, guess what we form? H2O. So this is where we get the cellular respiration equation. Glucose and oxygen combine together, or actually break together a lot, right? Form what? ATP, CO2, and water. Very cool. So that is the process of ATP synthesis in your cells. There's a lot of steps to it, but again, what did we need in order to do this? We needed to bring in our glucose through the symport. We needed to bring in oxygen through diffusion after we breathed it in and put it into our bloodstream. We needed the proteins in these membranes that we produced from protein synthesis. All of this is coming together, right? It all makes sense. I'm actually going to erase this board so I have a little more space. So if you want to take a picture, go for it now. All right, so next we're going to go through the cell cycle and some organelles. Right after that, however, we're going to go through an example of how these things interact to actually make one main mechanism of lowering your blood sugar very relevant to you. So let's start first with the cell cycle. You've probably learned this before. You've seen the little cycle of a circle, but I want you to know first off that not all cells really go through this process. In fact, the only main cells that will go through this are going to be your epithelial cells which we talk about in our tissue review video, because epithelial, this is the lining of your body, the cells that line your body, and they are highly abrased. They break down easily. We have to regenerate them constantly so that we can protect ourselves and cover our bodies, okay? So that being said, we need to be able to regenerate these rapidly. Now, other cells that can do this are going to be otherwise like stem cells also divide a lot. Sometimes whenever you have a wound, we're going to have rapid division of different blood cells. So for wound healing and repair, we have to basically divide our cells to get them back if they were damaged. Okay, so all of this stuff is very important in terms of regenerating our cells. But again, not all cells really do that. For example, your neurons don't really regenerate all that much. So that being said, we need to know the main steps of this process. So just look at this cell as compared to when I blew it up here on the screen. You see, obviously, the nucleus, that big dark blue circle, and then you've got other organelles inside. Now, as you notice, the first stage of any cell cycle is going to be going from this to that. Obviously, what did the cell do, children? It grew, right? And it's starting to multiply some organelles. So some organelles will start to divide, interestingly, get bigger, and that is called the growth 1 phase, shortened up as G1. So it makes sense. The first thing the cells start doing are just growing, right? However, once they're growing and they're at this stage, they're basically fully functional. They're doing what they need to do. But at some point, especially if they're an epithelial cell, they know, hey, at some point, I'm going to have to replicate myself. So the next thing we have to do in order to replicate ourselves is we have to have 2 of everything, essentially, right? If you're going to replicate something, you have to double everything. So what is it going to do? Well, it's going to start going through a process called S1 phase. S1 stands for synthesis, and not synthesis of just anything, but synthesis of DNA. We're basically going from 1 full set of DNA in a cell to 2 full sets of DNA in a cell. And that makes sense because the next cell will need all the instructions on how to build protein. So the way we designate that is we say at this point, before S1, this cell's nucleus has N number of chromosomes. That's going to be 46 chromosomes in the human cells, and that contains all the genes necessary for the cell to survive. Now, that being said, we're preparing for it to go from 1 to 2 cells. So once we synthesize all the DNA, we double it all. We say this cell's nucleus now has 2N. What the heck was that? 2N. So therefore, this will actually have 92 chromosomes inside of it. So it has double the amount of DNA that the cell actually needs, which it's not going to necessarily use it all. It's going to transfer it to the next cell here in a second. Now, once we obviously double the DNA, now what do we have to do? Well, we have to go from this big cell to an even bigger cell, okay? So that we have even more organelles that we can share with the next cell and then therefore share the other half of the DNA with. So obviously, as we are growing more, we call this phase G2, growth 2 phase, and we're basically going to 2 times the organelles, okay? So we're doubling the organelles generally speaking, not perfect number. So now we've got a 2N nucleus, we've got double the organelles, now the cell is ready to split and divide, right? So how do we do that? Well, the next step you've probably heard of before is going to be that M. So M is going to be called mitosis, which literally translating to splitting the DNA or the process of splitting the nuclear material. So how do we do that? Well, I want to set up a problem first, okay? Have you ever moved houses? So you had to move a bunch of big stuff out of the house, and the problem is you have very small doorways. So if you got your bed out of the house, you had to break apart the bed, make it really small, and then transport that bed out of the house. That makes sense, right? Well, think of the DNA. You have 6 feet worth of DNA inside of the cell before it divided. So therefore, inside of here, we have 12 feet, double the amount of DNA that we need to perfectly place 6 feet of it, so half the chromosomes over here, and the other over there. So each one has 46. Well, that's a lot of DNA, right? Long strands of DNA that we have to move from a microscopic cell to another. Okay, it would be the equivalent of taking out just everything in your house through one tiny little doorway. It's very difficult. So what we need to do first is take all the stringiness of the DNA and condense it. So I'm going to write right at the beginning of mitosis, the first thing we need to do is we need to condense the DNA. Think of it as like a bunch of 12-foot long string that we've seen, and we're going to compact it, okay? And we're going to compact it into something called chromosomes. So I'm going to zoom in here on the nucleus of the cell, okay, on all of these cases. And we will see something like this after we condense that DNA. We will see DNA in these big condensation sections like this. You've probably seen this before. These are your chromosomes. So for example, this is chromosome 1, and you copied it, right? So you have 2 chromosome 1s at this point. And you also have 2 chromosome 2s, chromosome 2, again, all the way up to 23 chromosomes, okay? So you have 92 total. So this is just condensed DNA. It's like you took that string of 6 feet of DNA and you wrapped it up into a tiny little condensed form, right? So why did we do that? Because it's going to be easier to transport it from one part of the cell to the next. So at this point, once the DNA condenses, we say this is the phase of prophase, okay? Prophase is when we condense the DNA into our chromosomes. Next up, we have to align it. So in this middle of the nucleus, we are then going to align those chromosomes in the middle. So it looks something like this. Okay, so we always say metaphase. This is M, metaphase. Remember M for middle. We're aligning them in the middle. It's called the equator of the cell. Alrighty. So when we align them, we're basically saying, okay, we've got the 6 feet of DNA right here. We got the 6 feet of DNA right here. We need to pull both of it opposite directions, right, to make it into 2 separate cells. So from that point, we will begin to pull them apart. And at this point, you may learn all about those centrioles. So there's like basically a little protein out here with some spindle fibers that will come and grab it on both sides, okay? So it'll look something like this. So it's basically like we're grabbing the chromosomes, we're going to pull them apart separately into 2 sections. How do we do that? Well, the next step is going to be anaphase. And I always remember this because the chromosomes themselves kind of look like an A. You'll see them kind of pulling apart in 2 different directions. So they kind of look like A's, all right? And that's because the spindle fibers are starting to pull them apart. The last main phase of PMAT, we always remember that, telophase, is going to be telos, means basically end, okay? So where are the chromosomes? Well, they are at the ends of the nucleus. So they're already over here on the opposite sides of the nucleus. And at this point, if we look at the whole cell, so I'm going to draw the whole cell here, we are going to begin to start seeing, I drew that a little wrong, let me draw it again. So it's all the way out to here. We're going to start seeing a little divide here, divide happening, okay? That's basically the cleavage for where we're actually starting to pinch it off. And eventually, the whole cell will pinch off, and I'm going to shrink the cell a lot. So we'll have 2 square cells after we divide it, and they will have those DNA, the full nucleus, all condensed with all the chromosomes again, or sorry, not condensed, but all loosened up with all the DNA necessary. So both cells will have N number of chromosomes. That's that 46, the healthy amount. And we've split, as I've drawn earlier, all of those different organelles inside of it that are gone from one to the other. So now both of the cells can begin functioning as normal, as brand new cells with the same exact DNA from the first to the second. Now this process of this cell dividing into those 2 sections, that is called cytokinesis. Cytokinesis literally translates to, you remember, cell movement, okay? So the process of cells moving apart from each other, turning into 2 separate cells. So that's the cell cycle. Once again, only common in those stem cells, epithelial cells, and whenever we're healing a wound. Now, as we mentioned, there are organelles inside of them. What organelles are we talking about? At the beginning of this video, I basically said, forget about this normal composite cell. It doesn't exist in your body. In A and P, these are the organelles that you'll probably see a decent amount of. So let's point them out. Obviously, we've talked about the nucleus with the DNA. In this orange, we've got the ER, stands for endoplasmic reticulum. Now, we have 2 main types. One, with all the dots, these are ribosomes inside of the endoplasmic reticulum. Anytime the ribosomes, the protein chefs of the cells, are in the endoplasmic reticulum, we call it the rough ER because it looks kind of rough, it's dotted. Whereas down here, we have something called the smooth ER, alrighty? Now, it makes sense that we'd have this because the ER helps to basically package and fold proteins. Package/fold proteins. So as the proteins are being produced by the ribosome from the mRNA, we're able to basically fold them in the right manner to make them the right shapes while we are in the endoplasmic reticulum. Now in the smooth ER, this is primarily for two things: detoxification as well as lipid synthesis, lipid creation. Now these are especially prevalent in your, think about it, liver cells. Your liver cells have a massive amount of both smooth and rough ER because they have to detoxify substances that are in your blood. They also have to produce cholesterol for your blood. So it makes sense for them to have more of this structure. Not all cells have this smooth ER. Now over here, we've got the Golgi apparatus. We probably won't talk about this too much. Golgi apparatus, okay? One connection here is once these proteins are made in the endoplasmic reticulum, they can be trafficked or sent over into the Golgi apparatus. This is where we can add different carbohydrates to them. So as an example, I talked earlier about how we produce these ID tag proteins on the outside that oftentimes have those purple, wherever my purple went, is in my hand. It's gone, the purple's gone, it's fine. Oh, it's in my hand. Told you I'm colorblind. They have those glucose attached to it, and these are the ID tags, but this was a carb. Well, the carbohydrate came from the Golgi apparatus. We added that carbohydrate to it so we can form that ID tag protein, okay? So very important. We also have these proteins called the cytoskeleton. So these are called cytoskeletal proteins. And it makes sense, these guys are holding up the cells. These are especially common in the neurons. Remember those long signaling cells? We've got to keep that shape proper. We do that through those cytoskeletal proteins like microtubules. But then these guys, we talked about the mitochondria earlier. Don't have to go through that. That's your site of aerobic cell respiration. So we got mitochondria. And then we also have these vesicles. You will see, I will tell you this, a lot of vesicles in different cells, in neurons, in a variety of epithelial cells like glands. And these vesicles are basically enclosed membranes. So this is like a membrane of the cell, but inside of it. And they usually contain some sort of a product. So as an example, if it was a neuron, this product inside the little capsule of the vesicle could be, as an example, a neurotransmitter, something like serotonin or dopamine, okay, neurotransmitter. It could also be like a hormone, like insulin, okay, and we'll talk about insulin here in a second as an example. But then we also have these vesicles that have embedded proteins in them. So I'm going to write embedded proteins. Now, where would this come from? Well, we know that some cells have membrane proteins on the outside, right? And so they'll let some things in if they're like this. Well, how did they get there in the first place? Well, there's a vesicle that merged with the membrane and basically fused those channel proteins into the membrane itself. So you'll see a lot of these happening in A&P. And again, they're just organelles packed with some product, some protein potentially, and we're putting them in different parts of the cell to make the cells work. Now, let me give you such a wonderful example. All right, so to beautifully give you an example of A&P when it comes to cell biology, we're going to talk about two cells and how they can interact with each other to maintain homeostasis. We're going to talk about the beta cells of the pancreas. Then we're going to talk about the liver cells, the hepatocytes, okay? Now the beta cells, we know, can respond to high blood sugar levels. So they're going to respond to high blood glucose. Now, we know that glucose is a fuel source. It's really good, but if we have too much of it, it can actually damage our blood vessels and cause all sorts of havoc, as we see in diabetes. Now, when they respond to it, the way they respond is they will release insulin, which is a hormone. Okay, it's going to travel in the bloodstream, and it's going to eventually lower the blood sugar. Okay, lower the blood glucose. So think of that, that's the goal. Okay, we're going to make insulin, it's a hormone, it's going to travel in the blood, change our blood glucose somehow by lowering it. Whereas on the liver cell side, these liver cells are very good at uptaking and storing extra glucose. So uptaking and storing extra glucose. So you can predict that if we have high blood sugar, high blood glucose, where are we going to store that extra blood sugar? Well, we're probably going to do it in the liver cells. But how do they know when to do that? Let's check this out. So combine it all together. We know that the beta cells release insulin. That's a protein. So let's draw inside of this cell these vesicles that are going to contain that protein hormone, and that's going to be our insulin. Okay. Now we know we need to release that when the blood glucose is too high. So let's show the blood glucose getting too high. So here's a lot. We'll draw a purple circle. Here is our glucose. It's really, really high. So I'll point at that. This is our glucose. It's too high. Well, we know that the beta cells are able to detect the high blood sugar. How do they do that? Well, in their membranes, they have a GLUT transport protein. So I'm going to draw that here. This is going to be a GLUT, okay? Literally translates to glucose transport protein. And it is actually something called a uniport. Okay, so a one-way directional of glucose. So what will happen is glucose will begin to come into the cell. Okay, so the beta cell is actually uptaking some glucose here, but it's unable to store it. So what does it do if it's unable to store the glucose? Well, obviously, we're going to go through all the process of glycolysis in the Krebs cycle, in the electron transport chain. And we know, you guys are smart, we've already learned about this, we're going to make a lot of ATP. So this beta cell is all of a sudden like cranking out a bunch of ATP. And it's like, holy crap, this is a lot. We must have a lot of blood sugar. So what does it do with some of these ATP molecules? Well, check this out. Through a bunch of cell signaling processes, what we do is we take these protein channels, okay, and these are actually potassium channels, all right? And if we know about potassium channels, we know that potassium is high inside the cell. So normally they would be flowing out, right? So potassium's flowing out, and it keeps the cell very, very negatively charged. Remember, cells have a charge to them. So what's happening? We have these potassium channels. We're losing potassium. It gets very negative. We know that cells, when they're negative, they do nothing. So they're doing nothing. They're not releasing insulin, which we need to release. So what do? The high ATP goes through several processes of phosphorylation. I'm not going to go into them, but it's actually going to close these channels. So eventually, high amounts of ATP is going to shut up these channels. Okay, it doesn't actually look like that, but that's what we're going to say it does. When that occurs, potassium no longer leaves the cell, right? So now potassium's building in the cell. Well, we know if we build up a lot of potassium, positively charged channels, or ions, the cell's going to get positive. When we get the cell positive, you guys are smart. Well, we can open those voltage gates. Remember that? So inside of this insulin cell, we will have these things that will open, and these are called voltage-gated calcium channels. So I'm going to point to these, voltage-gated calcium channels. Well, what happens? Well, the voltage, the higher voltage, because the potassium's staying in, opens these channels up. What are they? Calcium channels. Calcium then is always high outside of the cell, and the calcium will flow into the cell rapidly. Well, what do we know about calcium? Well, if you learn about the muscles, the nervous system, you know that calcium, I want you to remember this, likes to move stuff. Calcium likes to move stuff through a variety of processes. One such thing calcium likes to move is, boom, these vesicles. So what will happen is when the calcium flows in, it goes to these vesicles filled with our insulin, our hormone, and it will push them out of the cell, embed them in the membrane, and insulin will get released into the bloodstream. Right? Now, we could step even further back. How did we make the insulin? Well, the DNA, the insulin gene, right? The insulin gene inside of this nucleus was read. It was transcribed onto an mRNA transcript. That mRNA went to the ribosome. We formed the ribosome, the insulin protein were produced, and then the endoplasmic reticulum actually bubbled them off, made a vesicle, found its way there. And now, when glucose was high, check it out, it went into the cell. A lot of ATP was made. We closed with the potassium channels in response to that. That, therefore, made it positive. Opened up voltage-gated calcium channels, so calcium flew in, and calcium likes to move stuff, so we push the insulin out into the blood. It's crazy, but that is why cell bio is so important to understand how these processes work, right? So let's keep following it. Insulin is now in the bloodstream. Insulin by itself does nothing to the glucose levels. It does nothing. So insulin and the glucose are just going to be flowing through the bloodstream willy-nilly, just as if nothing is going on. But who do we need to go talk to about storing this extra glucose? Well, we know we need to talk to these liver cells, right? So check this out. In the liver cells, we have a protein that was made by the liver cell, and this is called a G protein coupled receptor. G protein coupled receptor, I am a nerd about these. They do so many things for your cells. And as you can see, there's a receptor aspect out here that can actually bind this insulin. So insulin's going to come and bind to this G protein coupled receptor. When that occurs, this G protein, which is labeled here, this is the G protein of the G protein coupled receptor, will pop off and get activated. So I'm just going to draw this guy, gets activated. Alrighty? Now, when G proteins get activated, they can do a variety of downstream cell responses. So what is this G protein going to do? Well, we know that it is going to go and open up, check this out, calcium channels again. So I'm going to open up calcium channels. Calcium channels, open. We know calcium likes to move stuff. Well, calcium, as we said, high outside of the cell always. So high calcium flows high to low into this cell. So we've got calcium, and it's going to move something. Well, what's it going to move? Well, we saw in the previous section of this video that we have some membrane proteins that are embedded, vesicles that are embedded with membrane proteins. And we have a vesicle like this in our liver cells. This is a vesicle packed full of, once again, GLUT transport proteins. So check this out. We bring calcium into the cell. That GLUT protein was produced by protein synthesis. The calcium's flowing in, pushes these vesicles out, and now allows more of those GLUT transport proteins into the outer membrane. Now, we could draw them similarly, I probably should have, as those guys in the beta cell. So here we go. We've got those GLUT transport proteins that were embedded into the the liver cell's membrane. Well, obviously, if we put more transporters into the membrane, where can glucose go? Well, obviously, it can go into the cell, this process. So now, all of a sudden, glucose is flying into these liver cells, and what happens to the blood glucose? If it takes out of the blood sugar drops. But where did the blood sugar or the glucose go? Into the liver cells. And now the liver cells will be induced to do an anabolic pathway using a variety of enzymes to take all that glucose and to start chaining them together to form these molecules called glycogen. Glycogen is basically stored carbohydrate that we can then, if we have low blood sugar at some point, chop it up, throw it out into the bloodstream, and raise our blood sugar. It is such a beautiful process, all mediated by the processes of cell biology. So if this was helpful, again, like the video, subscribe to the channel. If you want to check out other review videos like overview of chemistry or tissues or A&P in general, please check out the rest of my channel.